各位教授、同学、观众们，大家早安！我是今天的主持人，就读国立台北科技大学资讯与财经管理系硕士班的张兆怡。欢迎各位教授、同学们来参加由好奇学生大家问、大同大学、福音科技大学、文化大学、实践大学、动物大学、民传大学、静大学、辅仁大学、台北科技大学、慈济大学、嘉义大学、阳明高中联合主办的好奇学生大家问连接台湾系列专题讲座。谢谢现在线上参与的各所国内外学校学生，也感谢业界人士各位伙伴的热烈响应。大家知道好奇学生大家问的主旨是什么吗？好奇学生大家问的主旨是建立一个活动平台，邀请海内外专业人士，透过每月一次的专题演讲，将他们的知识经验与大家来做分享，让同学能够聆听前辈们的演讲，激发同学们的好奇心，自主学习并提出问题。那线上的同学们知道好奇学生大家问的宗旨是什么吗？当然，提问才能成就非凡的未来，所以我们愿意提供丰富的提问奖金，也希望各位同学踊跃的来做参加。那活动的时间和呈现方式呢？活动安排在每月第3周的星期五早上10:10到11:45，由线上视讯的方式来做呈现。那学生们又该如何参与呢？演讲前大胆提问，追踪我们的官方社群连结都在聊天室。那在 KKTS 报名活动时可以提问问题，演讲者会参考提问内容，规划本次的演讲，以及在演讲中填写问题表单，让演讲者在 Q&A 环节回答问题。那欢迎和感谢时可以按荧幕右下角的回应来鼓掌和谢谢。那演讲结束后，也可以参与好奇学生大家问的工作团队，学习团队合作能力。我们先请我们的节目主办人刘登凯博士为同学们介绍提问奖金的部分，有请刘博士。
好，感谢我们的主持人赵宇同学。那在这里，我先感谢我们的主讲人听卢博士，非常感谢你接受我们的邀请。我们感谢我这个数位时代的科奇将今天的主讲人介绍给我们，更担任今天的这个主讲人的介绍人，非常感谢你科奇啊。当然，我更感谢各校的老师同学的参与。那回到奖金部分，我们按照惯例，我们奖金部分分成2个部分。第一个部分是事前提问题，我们这次收集到非常多的问题，而且问题非常好。那么我会在当中选出第1名、第2名、第3名。第1名我给1000块钱台币，然后500，然后300台的奖金。然后这个第2部分呢，就是我们的卢教授演讲完了以后呢，同学会提出来问题，那卢教授会当场会回答你们。那这个部分我也选出3名来，那有这个第1名是1000，第2名是500，第3名是300所以我们这是2个部分。那这个同学们听到我宣布得奖以后呢，我们会有一个这个 QR code 让你去上网把你的这个信息登记起来，那这样我们就以便我们发奖金给您了。好，那我们等一下这个赵毅同学还会再介绍一下这个过程。感谢我们的主持人，那我把线上交还给你，赵毅同学。
好的，谢谢刘博士的说明。接着在这里跟各位同学讲解一下节目的流程。我们会在10:50结束演讲后，直接开始47分钟的 Q&A 活动环节。如果听了演讲者精彩的演讲后产生任何的疑问，欢迎透过聊天室的连结发送问题，我们将现场选出3个好问题给予提问奖金，也会选出演讲前报名表单中的3个问题，所以总共有6个得奖名额哦。问题可以包含卢冠达教授的专业领域、团队合作、心路历程等方面来询问问题。那请得奖同学活动结束后留下来加入颁奖的赖群，确认进入得奖赖群或是有联络上的信箱再离开，以利后续奖金颁发作业哦。本次演讲主题由现任美国麻省理工大学教授兼 Senti Bio 的创办人卢冠达教授为我们演讲寻找通过工程理解生命。那让我们欢迎数位时代创业小聚的国际主召集人柯奇来介绍今天演讲嘉宾卢冠达教授，有请。
嗨，大家好，我是数位时代的柯奇。今天很高兴邀请到国际级的创业家及生物科技专家卢冠达教授 Tim 来到好奇学院大灾问讲座。之前我们有幸采访过一次 Tim，然后 Tim 深入浅出的介绍了合成生物学以及他的创业经验，都让我们整个团队都获益良多。那 Tim 有双博士学位，分别是麻省理工学院电机与生意工程博士及哈佛大学医学院医学博士学位。在生物制药和合成生物学领域的研究跟创业活动都享有盛誉。Tim 的研究和创业活动跨足了不同领域，包括电机工程、合成生物学跟医学。那研究又聚焦于使用工程方法来改变基因、研发基因治疗和细胞治疗等新兴的领域。那 Tim 同时也是知名的连续创业家，和伙伴共同创办了8家生技公司。最新的 Senti Biosciences 也在2022年在美国纳斯达克上市。那我想说，我就不再多说，直接把时间交给 Tim 来分享今天的主题：寻找通过工程理解生命。欢迎 Tim。
非常谢谢，感谢大家的时间，邀请来 share 一下这个 screen 好了。Okay, great. Well, yeah, again, thank you so much. I'm going to present in English, but we can answer questions or talk about any topics later on in Chinese if it's helpful. So my name is Tim Liu. I'm an engineer by training, and my passion in life is trying to understand life and how does biology work. And the way we go about doing that is actually by engineering life. If we can figure out how to change cells or how to program cells, then that means that we actually have the understanding of how cells actually work. And so that's really been the focus of my research for the last 20 years or so, and also an area that I'm very passionate about, turning these into real treatments and diagnostics for human applications. So, where you guys are in Taiwan, you know the importance of the semiconductor industry and Moore's Law. Moore's Law basically was named after Gordon Moore, one of the founders of Intel Corporation. And what he predicted is every year or every 2 years, you could double the number of transistors on a chip. And that has led to an amazing explosion of computational power in the semiconductor field. The same thing is happening in biology. Actually, over the last 2 decades, the ability to read DNA through DNA sequencing and the ability to write DNA through DNA synthesis have also been improving at an exponential rate. And so if you can see here, the cost of sequencing a whole human genome, especially in the last decade, has dropped very, very quickly. On the right-hand side, with new technologies, we can print large amounts of DNA on a chip. And we can take that DNA and use it to engineer living cells. So what this means is we are now in a world where we can try to understand life by programming life. So we're no longer stuck with the traditional genetic engineering where we can only cut and paste DNA from one place to another. We're actually now able to program DNA. Every single A, T, G, and C inside of the cell, we can program and we can control it. And that's leading to a really amazing time in the field of biology and also in the field of drug development, where traditionally the way we discover new drugs is by chance. We made the original antibiotics were found by digging up soil or digging up sort of random samples and then screening for new antibiotics that could kill bacteria. Nowadays, we can actually use computation and we can engineer cells, we can engineer proteins, we can engineer drugs that are designed to target very, very specific mechanisms using computational design and using synthetic biology. So this is a really exciting time for people who really are curious about how life works. And it's a time when a very interdisciplinary background, people who have computational expertise, people who know how to do machine learning, people who know about biology or medical physicians who have an understanding of how diseases work. We can all come together and work as teams to try to uncover how disease works and also to design better diagnostics and better therapeutics. So today I wanted to share with you 3 examples of the use of synthetic biology combined with machine learning and computational approaches. Number 1, to understand how cells work. And then I want to share a couple examples of how we can actually design novel drugs and novel detection mechanisms. So one of the key challenges in biology today, and actually ever since the last 50 years of DNA and how we understand how DNA works, is understanding how genes control biology, right? So, for example, I have a different DNA than you. Why do we look different? Why do we have different heights? Why do we have different biological properties? And so it's very hard right now to know, why one or more genes, how it impacts how biology works. And so almost all of the work over the last 20 plus years in the biology side has been trying to figure out the linkage between the DNA inside of us and the final biological behaviors. The key challenge in this space, which is called genotype to phenotype relationship, is that biology is very complex and it's very multifactorial, meaning usually it's not one gene that causes one thing. There's no one gene that tells you how tall you're going to be. It's usually multiple genes together interacting with each other to give you a function. However, this is a big challenge if we want to try to discover and develop more precise medicines. And so what I would argue is, in order for us to create better, more precise medicines that treat disease more effectively and reduce the likelihood of side effects, number 1, we need much more powerful ways of mapping disease. We need to understand what causes the disease, first of all, and it's usually not just one cause. Usually it's multiple interacting genes or proteins that are responsible for that. We also need to precisely design therapies to target those mechanisms, and hopefully that will allow us to have better impact for patients. So what's the big problem? The big problem is biology is very complicated. So for example, imagine you have about 20,000 genes in the human genome, and if every single gene interacts with every other gene, in many cases, then it's 20,000 times 20,000 different possible combinations. But we know that actually genes interact with more than just one other gene. If you look at, I'll show you a picture later, genes can interact with, or proteins can interact with 3, 4, 5 other genes. And so it's a combinatorial, very complex web of interactions with many, many different possibilities. And so there's no way right now we can screen through comprehensively all possible, trillions and quadrillion different combinations. Less than 1% of this space has been explored today. And so we need new technologies to understand how this network works. And we also need computers to help us predict and make sense of how these networks work, because it's too complicated for humans to understand by ourselves. So, if you study traditional molecular biology, probably what you learned in middle school or in high school, is over the last 50 years, we spend most of our time looking at one gene at a time. We say, okay, what does that gene do? People have great papers, great discoveries on one gene at a time. But in reality, biology is actually in networks. It's not just one gene, it's a network, just like the internet is a network. And that network is very complicated. Here's a picture of protein-protein interactions that have been mapped in yeast as an example, and just showing all of the interconnections between different pairs of genes and proteins inside of the cell. There's over 200 million unique pairs, over 1.3 trillion unique triplets. And if you think quadruplets or quintuplets, and you keep increasing the scale of this, it sort of blows your mind. It's very difficult to explore this. So to try to tackle this problem a few years ago, in my lab, we tried to develop some new technologies to understand and pick apart these networks. So this requires 2 things. One is that you can go in and you can turn on or turn off multiple genes at the same time, right? So we want to turn on gene 1, turn on gene 2, turn off, and see what happens. But if we do that one by one, it's going to take forever. It'll take more than my lifetime to try to do that. So we need a way of doing this in very, very high throughput. Number 2 is, once we have that data, it's again impossible for you to just look at this diagram and understand what's going on. You need computational techniques to really help you understand and crunch the data. So again, we have developed very high throughput systems that leverage computation and very high throughput biological experiments to basically go in and knock out or perturb multiple genes at the same time. So instead of just going in and doing one experiment with gene 1, and then another experiment with gene 2, and then another experiment with gene 3, what if we could do 100,000 experiments at the same time? And we could actually look at what happens when you knock out gene 1 and 2, or gene 1 and 3, and gene 1 and 4, and 1 and 5 together. So we've developed technologies that allow us to perturb this in very high throughput. This is using a technology that recently won the Nobel Prize called CRISPR, where you can basically go into a cell and actually knock out multiple genes at the same time. And it's very easy to do this. So what this has allowed us to do is actually look at interactions inside of the cell and identify important pairs of genes where one gene, if you knock it out, will affect the other gene. And actually recently, a few years ago, we spun this out into a company that could use this technology for drug discovery. This type of company is called Senti Biosciences. And I'll walk you through why this is potentially very powerful for understanding drug development. So what Engine Bio is doing is looking to develop more precise cancer therapeutics. So a patient who comes in who wants to be treated, the problem right now is if you just give people chemotherapy, it's very toxic. And chemotherapy will kill off the healthy cells and also the cancer. So you get very sick even though you're trying to treat the cancer. So we want to try to develop a much more precise set of drugs that only target those cancer cells while limiting the effect on on healthy cells. And the way we do this is, number one, we collect a lot of data. So we collect, extensive data from clinical data sets, internal data sets, the literature. We use natural language processing, so basically automated machine learning techniques to read all the literature that's out there. And that allows us to assemble an initial database telling us what genes potentially are interacting with each other. Number two, what we use is we have a computational algorithm, and I'll show you some more detail of that, to predict, okay, if I go into a patient that has this mutation or this gene that has a problem in cancer, then I should use this drug to treat it more effectively. And then what we can do is use our experimental platform to validate this. So we can rapidly screen hundreds of thousands of different possibilities in one single experiment. And because we have this computational platform, we can increase the hit rate by 20 to 30-fold as well. And as a result, the idea here is in a complex network, each one of these dots is a different gene. For example, we can knock out different pairs or different triplets of genes at the same time and see what happens. So I mentioned earlier, one of the key areas that is really transforming biology today is the ability to use machine learning to understand biology. So even though humans like to understand how biology works by starting from genes, then predicting proteins, and then predicting higher order interactions, it's very hard to build a purely rational and mechanistic model of how cells work using this sort of approach. Maybe one day we'll achieve that, and that will be a great breakthrough. But as of today, we don't have the computing power or the data sets to really build a very accurate model from the ground up. The most accurate model that's been built was published a few years ago for mycoplasma, a very, very small bacteria, required a ton of simulation time in order to make predictions. So instead, where the field is going today is actually to develop machine learning methods where you can take these large data sets, for example, understanding the genes, the proteins, the metabolites, and then develop essentially a model to try to explain how the biology works. Now, there are many still challenges in this space in applying machine learning to understand biology. The data that you collect from different labs, they might be a lot of different quality or variability. And so for Engine, at least, we've focused internally on doing a lot of QC to make sure that the data that we're using to train these algorithms are very, very high quality. Number two is machine learning by itself usually is a black box. It doesn't give you an explanation of why something happens. It just gives you a prediction that something can happen. And so for us, we not only use the machine algorithm, we also do experiments to really understand why it happens. And finally, we just need to really do this in a context-specific way and do this very quickly. And so fortunately, having a very powerful computational team, but also algorithms that are optimized allows us to process a lot of this information. Why is this important, right? Number one, we want to understand how biology works. But number two, this actually is very useful for treating patients. What this allows us to discover is something called a biomarker. So a biomarker is something that you can identify inside of the cell. For example, a mutation in a gene inside of a cell. And if that patient has that mutation, and we use a very specific drug as a result to treat that patient, we can significantly increase the clinical success rate. So for example, this is a study done and published a few years ago, looking at the probability of success in treating patients with different types of cancer if you have a biomarker. And if you have a biomarker, on average, you have about a 13 times higher successful treatment rate versus no biomarker. It helps you de-risk the target and accelerate the drug development path. And ultimately, it's better for patients because they're not treating these patients with drugs that don't work. So one of the ideas here that we work on is basically creating predictive biomarkers for drugs. So for example, in a normal cell that has both gene A and gene B, you treat with a drug and hopefully these cells still survive, so you don't kill those cells. However, in a cancer cell where there's a mutation, for example, let's say there's a mutation in gene A, that becomes your biomarker, right? That's different between the cancer cell and the normal cell. And as a result, if you apply the right drug, the drug should kill those cancer cells because they have the biomarker. The cool thing about, nowadays we know that in cancer, at least there's about 3,000 genes that have that are present in over 5% of all cancers, right? So there's a lot of potential biomarkers out there, but less than 1% of these genes have actually been associated with a precision medicine drug. So there's a lot of opportunity here to discover new biomarkers and make better drugs. And so let me just give you one example of this, but there's actually many more that you can read about and I can talk about later. So there are, for example, existing biomarkers that are used for the treatment of cancer. For example, there's a very large and successful class of drugs called PARP inhibitors. So if you have breast cancer or ovarian cancer, these PARP inhibitor drugs can be very effective. However, it's even more effective in patients that have a particular mutation in a gene called CDK12. And you can see here, this is a graph using breast cancer cells, where if you have the mutation, which is shown here in red, you basically have 12 to 17 times better killing of those cancer cells compared to patient cells that have no mutation. So therefore, if you're a patient that comes in, you sequence your cancer, you have the CDK12 mutation, you should take these PARP inhibitors because you're 10 times, your cancer is likely to be 10 times more sensitive to those drugs, as an example. Using our platform, our machine learning and CRISPR platform, we've been able to discover other types of biomarkers as well. And in some cases, we've been able to discover ones that are very, very powerful that give you 100 times to 150 times even better killing activity. So hopefully what I showed you through this part of the presentation is by using machine learning, by using CRISPR engineering, and by going into cells and being able to knock out many, many genes at the same time in this network, we can actually find biomarkers that actually help us design drugs that are much more effective for patients downstream. So that's the idea of using some machine learning and high throughput engineering to understand and find disease networks. At the same time, we can also use this ability to engineer DNA to create diagnostics and therapeutics. And that's a very exciting, I think, future field. So let me give you one example of this. All of us, I think, know that sometimes our gut, our stomach, our intestines, they change the way we feel, they change the way we behave. But we actually don't have a very good way of knowing what's going on inside of our body. You can't just look inside of your colon very easily. If you go to the doctor, the best way they have right now is to put a scope into your colon to take pictures, or to give you a pill that has a little camera that takes pictures as it goes from your mouth all the way to the other end. So what happens if we want to try to understand what's going on inside of our bodies, not just taking pictures, but also understanding the chemistry that's inside of our bodies? Well, one way of doing this is to use bacteria. So I'm sure all of you guys know that we have healthy bacteria that live inside of our body. So why not use those bacteria to understand what's going on inside of our bodies? For example, what if we can engineer a bacteria that goes from your mouth, you swallow the bacteria in your yogurt or your pill, and the bacteria goes down and then it detects bleeding or inflammation or infection, and it can actually report to the outside world, for example, through a wireless connection to your cell phone, to tell you that, hey, you're bleeding inside of your colon. You should probably go get checked out for colon cancer, as an example. Or alternatively, what if you could have the bacteria remember that it detected bleeding, and now when it comes out the other side, like in your toilet, it maybe change color. And that allows you to know what happened inside of your body because the bacteria tells you. To do this, we need to engineer that bacteria to detect disease, and we need a way for us to read out the signal. So we published this a few years ago, this idea that you could take a pill, that we engineer electronics onto the pill to basically detect light produced by bacteria. And the bacteria is engineered to produce light only when it detects, for example, bleeding inside of the gut. Then the chip also contains a wireless connection that can send a signal to your cell phone. And so on your cell phone, you can read, am I sick, essentially, in my gut? This is a project we did with our collaborators in the electrical engineering department. This is the pill initially. The first generation was very large. I don't think most people want to swallow this, but it worked. There was a photo detector where we put the bacteria. It detects light being produced by the bacteria. It has a very small battery, and then it can transmit this information through an antenna to your wireless connection. And what we did is we engineered bacteria, probiotic bacteria, that can detect blood, first of all. So this is looking at the electrical current coming off of the photodetection chip in the presence of blood. And you can see when blood is added, in about 30 minutes, you can detect a current coming off of this signal. We then put this pill into pigs as our proof of concept. And we basically tested the pigs in the presence of blood or no blood. And you can see again in the pig, you can detect the electrical signal in about 45 minutes to 60 minutes. So this is a demonstration that you can actually make a pill. That pill can, number one, take pictures, but it can also sense things like bleeding or other signals inside of the gut that are telling you that you have disease. Well, recently we published a paper that extended this in a couple of different ways. One is we made the pill a lot smaller. So now this is a size that, number one, won't get stuck in your gut, and also much more easy to swallow. But it's the same concept. We can put bacteria on the small chip. The bacteria can detect inflammation or other biomarkers of disease, and then it can transmit to your phone. We use this to detect inflammation in your gut. So for example, if you have inflammatory bowel disease or you have infection, your gut produces a lot of inflammation, and we want to detect that. So one of the molecules we use to induce gut inflammation is called tetrathionate, or TT, shown here. And we basically built a sensor to detect inflammation in the gut. And then show that, again, it worked in animals. So this is an example where we put the pill into the intestines of pigs. And we showed that, again, you could detect a photo detection current coming out of the chip in about 30 to 45 minutes, similar to before. And the sensitivity and the specificity of the system is pretty good. So we're very excited about that platform. We can use it now to diagnose what is going on inside of the body. And the next steps for that project are to work with clinical people, so doctors who study the gut, and try to run clinical trials with this product so that we can detect what's happening inside of their patients' bodies. The last thing I'll mention is actually not only can we use living cells to treat disease, to diagnose disease, but we can also use living cells to treat disease. And I think that's a very exciting, again, future for the field. So here, what we're thinking about is using living cells as a way to treat disease. Now, just as a reminder, basic biology, DNA turns into RNA, turns into proteins. Obviously, real-world biology is a little bit more complicated than that, but this is the basic idea. And so we can use DNA programming in order to make all sorts of new therapeutic modalities, including RNA, protein drugs, and living cellular drugs. And that's actually what's been happening in the field for the last 30 years. So instead of just using chemical drugs like aspirin or traditional medicine, we can actually engineer cells to make new therapeutic molecules. So the first example of this were monoclonal antibodies, where you can copy the antibodies that our body naturally makes, but you can use those antibodies to go after a different target. And that, again, requires a lot of genetic engineering because you need to change the DNA, and that DNA produces a different antibody. That has led to a field that has over $100 billion of global value. It's also allowed us to make really transformative new companies like Amgen, like Genentech, and others that are specializing in this area. In the last decade, though, there have been many, many new types of drugs that have been developed. One of the first types of drugs is called gene therapies, AAV gene therapies, where you use a virus. And with that virus, you inject it into the eye, for example, and it infects your eye cells, and it delivers a gene that can fix a bad gene inside of your body. And that's really amazing. It's a permanent treatment, and it can cure patients. In 2017, we had the approval of the first cell-based genetically engineered T-cell therapy. So T-cells are a special type of cell in your blood. And if you have cancer, specific type of cancer, you can go in, they take out your blood, they isolate the T-cells, they genetically modify the T-cells to do something called a CAR, or chimeric antigen receptor. And they put those T-cells back in your body. And those T-cells can hunt down and find cancer and kill those cancer cells. We also have RNAi therapies where you can use RNA, small RNA, you can deliver it into the body to knock out or knock down a gene that you don't want. So for example, if I have a gene that gives me high cholesterol, we can use this to knock down that gene and improve my cholesterol, as an example. And actually mRNA vaccines, which we all know about, for example, the BioNTech vaccines or the Moderna vaccines, allowed us to very, very quickly overcome COVID. mRNA vaccines are only possible because we can make DNA. So you make DNA first, and then you make mRNA. And the advantage of these types of vaccines is they're much faster to make than protein-type drugs, because once you know the sequence of the virus, you can make the mRNA vaccine very, very quickly. And that's what allowed companies like Moderna and Pfizer and BioNTech to develop vaccines very quickly. Very interesting side story. Moderna, actually, the initial technology that allowed Moderna to happen was funded by part of the US government about now 10 years ago. I was part of that program as well, and there were actually multiple approaches that the government was funding. They put about, I think, over $100 million US dollars into that program. It was all led by a very, very, very bright colonel in the US Navy. His name was Dr. Jeffrey Ling. And his idea was that one day there might be a pandemic and we need a very fast way to respond. And the traditional ways of making vaccines were too slow. The traditional vaccine for flu, for example, you need to grow it in eggs, and that takes forever. So he wanted to develop very fast new methods to develop these sort of vaccines, and he funded many, many efforts, including mRNA, to be able to do that. I just think it's very amazing that somebody had the foresight 10 years before COVID ever happened to invest in that high-risk technology to encourage the development of this platform, even though no one really had an application at the time. And I think that's one of the lessons I've learned is that, in science, it sometimes takes a long time for these technologies to mature. You don't exactly know when they're going to find the best application, but we need still to be bold and think about the future of what could make things transformative. If the future, what should the future look like? And let's try to make that, even if it requires a lot of hard work and time and investment to make that happen. Okay, so I'm going to tell you a little bit about cell therapies and what we're doing at Senti Bio to overcome the limitations of today's cell therapies. So the way cell therapies work is, again, you take a T cell or another type of cell called the natural killer cell out of the patient or out of a healthy person. You genetically modify it, and then you put those cells back, and those cells will go and hunt down cancer cells to kill them. In order to do that, they need to recognize a target on the outside of the cancer cell. So here's a picture on the left-hand side here. This is the T cell or NK cell. It contains a protein, which is this little green thing here on the surface. And this protein, this green protein, needs to recognize a green target that's on the cancer cell, which is these yellow cells. And if it recognizes that, then it basically kills those cancer cells. And so there are now approved products that, for example, this product called in 2017, this CAR T product, now made by Novartis and Kite, is basically using this sort of technology. However, there's two limitations of these sort of approaches right now. One is if you only go after a single target, you might kill most of the cancer cells, but if you have some cancer cells that don't express that target, you're going to miss them. Number two is, what happens if you have healthy cells? For example, these green cells are supposed to mean healthy cells, and those healthy cells also express the target, then you're going to kill those healthy cells. So that's a big limitation, and as a result, very few CAR therapies have been developed to date. Our hope is that we can program logic and computation into living cells to overcome this issue. So for example, can we program, just like you program a computer with logic gates, we can program these cells with logic gates as well. One type of logic gate is called the OR gate, which will kill any cancer that expresses A or B. So for example, if you have the green target or the blue target, we're going to be able to kill you. So we can design receptors that have two different heads. The green head targets the green target, the blue head targets the blue target. Nice example, or create a better killing activity. However, the other thing we want to try to do is also make sure we can protect healthy cells. So what we can do is engineer what we call a NOT gate, which is in this purple receptor. This NOT gate can recognize those healthy cells, for example, through the purple target, and shut down the activity of the product. So it says, do not kill those cells. So we've now done this. Let me just show you an example of this in action, which I think is very exciting. So this is a video of cancer cells and healthy cells. Cancer cells are in red, and the healthy cells are in green. And so we're taking pictures in a microscope over 7 days to look at what happens to the cancer cells and healthy cells. So for example, with no treatment on the left-hand side here, what do we see? Both the healthy cells and the cancer cells grow. In the middle is what happens if we only target a single target called CEA. CEA is present on both the cancer cells and the healthy cells. And so what happens in this case is you kill both the green cells and the red cells. And that's a problem, right? Because in patients, that means you're going to kill their cancer, but you're also going to kill their healthy cells, and you're going to cause a lot of side effects for these patients. However, if we have our logic gate technology, for example, we can engineer our CAR NK cells to kill only when you express CEA, but not if you express VSIG2. So VSIG2 is a healthy target. It's only found on those healthy cells, is not found on those cancer cells. So what happens here, you can see very clearly, is that the healthy cells survive and they keep growing, but the cancer cells die. And this is only possible because we've essentially engineered this product with the ability to recognize CEA on the cancer cells and not VSIG2 on the healthy cells. So what this allows us to do is build cell therapies that achieve very, very strong protection. So they have very little killing of healthy cells, which is in blue here, but they have very high killing of the cancer cells. And we get over 98% protection of this sort of product. This is not the only type of programming we can do in living cells. We are designing, just like, again, you can design an electrical circuit, we can design gene circuits using our DNA programming technology to do logic gating, to do what we call multi-arming. So you want to engineer a single cell that attacks multiple targets at the same time, to have what we call a regulator dial. We can turn on or turn off what the cell is doing inside of your body. So if the cell is too crazy, you turn it down. If the cell is not active enough, you can turn it up. So you have that ability to control your biology. And we can also develop products that have sensors that turn on only when they're in the right place. For example, they only turn on in the eye, but they don't turn on anywhere else. Or they only turn on the cancer cell, and they only don't turn on anywhere else. So this is all, again, possible because of the tremendous and exciting improvement in synthetic biology and genome editing and other sorts of tech and machine learning and other technologies. I think the last decade was really laying the foundation for a very exciting time in the future. We now have the Human Genome Project completed. We know the DNA that's inside of the human cells. People are rapidly characterizing as much data as they can about human biology, information about proteins, about RNA, about all sorts of different molecules. We have machine learning technologies that are allowing us to understand how these networks work. The FDA is becoming more and more open to these new types of drugs and new types of approaches. So just a bit of background, I started off as my undergrad education in computer science and electrical engineering. That was in 1999. But because this was right about the time when the Human Genome Project was being published, I got very excited that in the future we could program biology just like we can program a computer. And so I think that reality is now starting to come true. I think to me, this is the decade of biology when we're really going to, sorry, not the decade, but this is going to be the century of biology where we're going to figure out really how life works because we now have the ability to program life. And so I think it's a very exciting time for any of you in the field who are considering this as an area. It's an exciting time to get in and really apply your different types of expertise to solve these big problems. The other thing that makes this a very exciting time is that actually these breakthrough technologies will lead to new companies. This is not just a field where you can do academic research, although that's great if you want to do academic research. But it's also a place where you can make new drugs, you can make new companies, and we're going to hopefully bring new ways of treating disease to the market so that patients can benefit. Just in my own experiences in the past, being a professor, but also now working on startups and taking companies from early idea all the way to exit, there's many different challenges along the way. Certainly being able to do basic research and get enough funding to do that is really important. One of the big challenges actually is in the middle. Once you have the data, for example, we published a paper in a journal, but now what do we do with that information? How do we turn this into a real product? We need teamwork. This is not a place where one person can do it all. You need a team of people who are, very young and experienced and driven, young and driven scientists or inventors or entrepreneurs, coupled together with people who have been in the industry for a very long time. So they know how to navigate through the FDA, how to raise money, how to put together, clinical plans. This is the hardest part of the whole process. I think we call this the Valley of Death. It's really that early translation and proof of concept to get as much data in humans as you can. Once you have that, the downstream is frankly much easier. Being able to just execute on your clinical strategy, do partnerships, et cetera, that's all possible. It's really this early Valley of Death where we need people with expertise and drive to do this sort of thing. And so my personal opinion, it's an exciting time because there are many new types of transformative platforms and R&D capabilities that are coming to the table. We think about the use of machine learning, robotics, synthetic biology, allowing us to understand how biology works, but also how to treat it. There are many diseases out there that need new types of drugs. Certainly cancer, oncology is a big one. I think recently, now that we have some drugs that are approved to treat Alzheimer's disease, brain disease, also called CNS, central nervous system diseases, is a major area where people are very interested in trying to understand how these brain diseases work and try to treat them in much more specific ways. Traditionally, the drugs that we have to treat brain diseases are not very good, and they're not very specific. Another place where you can prove out your drugs is in rare diseases. So patients that have a very specific gene mutation, if you can go in and fix it, that's another way of generating good proof of concept. Immunology is another area. So if you have inflammation, liver inflammation, skin inflammation, et cetera, these are also areas where these new types of technology are coming to play as well. So I just want to thank you for your time. I'm happy to take any questions. I hope I shared with you that I'm very excited that the exponential improvements in our ability to read and write DNA will transform how we understand life and how do we tackle disease. Synthetic biology uses the ability to write DNA in order to explore the many complex networks that determine how cells function. We can also use this technology to detect and diagnose diseases in our body. And we can also use this technology to create gene circuits to create therapies for these diseases. I think there are a lot of really exciting academic breakthroughs and commercial breakthroughs that will happen when we as we continue to apply these powerful technologies to map and diagnose and treat biology. And actually to make this into a commercial reality will require a very integrated approach. People who have computational skills, machine learning skills, engineering skills, biology skills that work together to solve these problems. So it's an exciting time, and hopefully if I piqued your interest a little bit to get into this space, yeah, that'll be a good outcome. So thank you very much, and I'm happy to take any questions.
OK，非常感谢听入精彩的演讲。那我们现在要进入 Q&A 环节到11:37。如果现场有任何疑问，欢迎利用公众人员背板上的 QR code 或聊天室的 Q&A 表单连接来问问题。那问题被挑中，线上的参与的观众可以开麦克风与讲者来做互动，追问议题。如果没有问题，也请说谢谢听录。那实体现场提问的同学，听到我喊到你的名字的时候，请到台上拿麦克风，站在镜头前与讲者互动。互动当下可以追问议题，如果没有问题，也请说声谢谢听录，并回到座位。那我们现在就要准备播放第一个问题了，稍等我一下。好。第一个问题由北科大机械系的谢承彦同学，他问：请问您是如何使用工业半导体的机体电路概念来建构基因和细胞疗法的呢？最初又是为何有这个想法？因为对这个技术感到非常新奇。
Okay, yeah, 这个是很好的问题。So, I think I'll answer this in English if I can, but I think the reason why we take this idea is because we've learned so much about how to program silicon and program computers, right? There's a whole framework that allows us to take this very simple transistor and then scale up to very, very complex computers and networks. And to do that, every single level, the key innovations that are required is that we need to understand the system, create modularity, and create interfaces that allow us to scale. That's the only way we can solve that sort of scaling issue. The problem is biology is not modular. It's not designed by a human. It's designed by evolution. That's why the network is so messy. If you look at that picture I showed you, right? Because there's no person who designed that. If you were to design that in your class, you would fail because no one can understand it. So the problem is we're taking a system that has been given to us by evolution, and then we're trying to figure out what do we do with it? So if we want to engineer that sort of system, we need to make it simpler for ourselves. And we can't remove all possible interactions, but we can do things when we design these gene circuits to make them more modular. So for example, we can try to design genes that only have interactions with a very specific location in the genome or other proteins. And so we don't have to worry about everything else that's happening inside of the cell. So I think one of the concepts that we're adopting from traditional mechanical electrical engineering, this idea of modularity and abstraction into biology, is just to make our life a little bit simpler. Now, like I said, it's not possible to do this 100% because biology is messy, right? If you think about a cell, all these genes, all these proteins, it's basically in one place and they're all bumping into each other, right? It's not a chip. So number one, we spend time trying to make this as modular and as abstract as possible. But then two is with the new improvements in machine learning technologies, even if we're not 100% perfect at isolating each element, using some of these machine learning methods, at least we can map out the consequences of those interactions and try to minimize them.
OK， 请问谢承念同学在吗？麦克风没有开哦。
OK，
有要追问问题吗
没有了，谢谢，谢谢教授。
？OK，
好，谢谢。
谢。那我们再换下一个问题，由国立台北科技大学光电工程系的林宇恒，他想要问，请问如何改变现今大众对生命工程的态度？像是基改农作物，基改农作物相对于一般作物可以有更好的收成、更丰富的营养，养活更多人。而我观察现今台湾大众却普遍对这种生命工程保持反感的态度，如何翻转台湾大众现今对生命工程的印象？
这个其实是个很好的问题，这是全世界很困难的一个问题，因为其实很多人针对这种 genetic engineering 这种技术，好像有一些不是很清楚而了解的一个基本的概念。那 the way I approach it right now actually is I actually tell people, look, you know, if we didn't have mRNA technology, we would all still be stuck with COVID right now. Right. And so that's just one example. Genetic engineering can be very good for us. You know, we have new drugs, we have mRNA vaccines, we have antibodies. You know, our lives have improved because of this technology. Now, of course, people are concerned that if you change, maybe you change your food and you eat it, some people think that it causes bad problems. And actually, the issue I think here is really about education. I think we need to educate people earlier on, like, you know, in middle school and high school, about what genetic engineering is. And, you know, I go to Taiwan a lot, right? I know the fruit there is amazing, right? That fruit is amazing because we breed fruit for really good, sweet taste, nice taste. What's actually happening when we do breeding? We're actually changing the DNA inside of the fruit. It's just that we're changing it in a very traditional way. So all genetic engineering is doing that in a targeted fashion. And I think we need to explain to people what the risks and also the benefits are in a very logical fashion. I think that's probably the only way that's possible. But I think this is a big problem in the whole world about science, right? Some people don't believe in climate change, even if you try to explain to them the science of climate change. You can spend a lot of time explaining to people why genetic engineering is not something to be scared of in most cases. But some people just have, you know, they're scared by things. So I don't know if you can convince everyone, but I think we need to try to use science and early education and really use rationality to explain these things. That's my personal opinion.
OK，请问林宇恒同学在吗？
要做网站，在网络站。你在吗 ？You? Yeah, no any problem. Let's take after two.
谢谢。
OK，谢谢。那我们再换下一个问题。北科大光电工程系的吕正豪同学，The importance of interdisciplinary collaboration. Interdisciplinary collaborations are crucial in the advancement of synthetic biology.
Yeah, so this is very important. I think, you know, a lot of science in the future is not going to be done by one person already. I mean, already we see that many big scientific projects require teams. Certainly, even in academia, where traditionally a PhD is just like one person, you lock them up in the lab and they just do their thing for 5 years. But that's not the way I think most science will be done in the future. And if you look at companies, one of the big things I learned as I got into this industry world is a lot of science is done by teams because it's more effective to do that. And especially today in biology, I think interdisciplinary experience is very important. I don't think any one person has all of the know-how to be able to solve these big problems. Let's say, I want you to tell me what causes cancer. That's such a big problem that you can't solve it just from one way, right? You need people who know how to do the biology, right? Know how to do the experiments. You need people who know how to interpret those experiments. You also need computational algorithms because there's no one person that can understand everything. So you need people who can program, let's say, machine learning or a model, computational model, understand it. you need doctors who can take that information and hopefully test it in humans. So I think all of these expertise, we all come up with from different backgrounds, but I think the importance is a few things I've learned in my career is, one, you have to be willing to work with other people, right? Because I think sometimes if you just want to chase your own credit, that it limits what you can do, right? So working with people is important. Two is I think you have to learn how to communicate with people in different languages. So if I'm a biologist by training, I might not understand deeply what the machine learning person is doing, right? I don't know how to code, for example, but I need to understand how to talk about the outcome of that code, right? I need to have enough shared language that we can actually have a meaningful dialogue. So I think that means is, in your education to really try to expose yourself, even if your primary major is in one area, like electronics. If you're interested in the space, take a biology class. At least you don't need to be the expert in biology, but you need to learn the language of biology. Or if you're a biologist, maybe take an introduction to programming class. It's a pretty useful skill, and at least you'll learn the language and understand some of the basic ideas. That way you can actually communicate once you're out in the real world. That is very valuable for your own personal career to have that skill set. But also I think if you want to, make these sort of, be a leader in this space or explore these sort of breakthrough ideas in the future.
请问吕正豪同学在吗
No further questions.
？OK， 好，谢谢你。那我们再换下一个问题。Real-world applications of recent advances. Can you share some of the most exciting real-world application of current synthetic biology?
Yeah, sure. So I think synthetic biology is a big term, but I think I classify synthetic biology as any sort of new genetic engineering approach. So I actually think like AAV gene therapies, CAR T-cell therapies, mRNA vaccines, these are all examples of sort of what synthetic biology can do, right? So within 1 month of COVID-19 being sequenced, we had the first prototypes of what an mRNA vaccine was. That's amazing, right? No, I don't think that speed record has never been achieved before. But I think that's a very clear world application. I think right now there are a lot of drugs in development. There's, I think, over 100 different types of cell therapies in clinical development now in phase 1, phase 2, phase 3 trials to treat all sorts of different cancers. There are hundreds of gene therapies out there as well that are in clinical development. And actually the first CRISPR therapy just got a positive review this week. So I think that technology is going to start making its way into humans. So I think these are all examples where in the future, we're going to be able to modify DNA, write DNA, and treat diseases in very, very new ways. And that's for treating cancer, for treating rare genetic diseases, for treating blood diseases, all sorts of diseases that right now you just have to suffer with it, or you might be born with it and you have to live with it for the rest of your life.
Professor, thank you for your advice, and I think those are good advice, and thank you for sharing it with me.
Thank you.
OK， 谢谢。那我们再换下一个问题。再来是台北科技大学机械系的林旭维，他问：对于合成生物学未来的发展，你有哪些预测或期望？这个领域在未来10年内可能会出现哪些重大的创新？
Yeah. The one area that I'm actually the most excited about right now, and I talked about this a lot today, is really using AI to predict how biology works. Because I mean, I've been working in this field for 20 years. When we first started, we really wanted to try to build bottoms-up models to predict how the cell works, right? Can I just simulate everything inside of the cell? And I think what we realize is doing that is really, really difficult. It requires too much computational power. Like unless quantum computers get invented and scaled up, I don't think that's going to be possible in the near term. But using these new machine learning technologies that have been developed over the last 5-10 years, large language models to understand DNA, protein, all these are just sequences. They're just language, right? DNA is just a language, protein's a language, RNA is a language, just like ChatGPT understands English. We can use these methods to understand how biology works and predict how biology works. So I think we already have a lot of good evidence for proteins. You know, the Lasker Award this year went to a technology called AlphaFold, developed by the Google scientists, where if I just give you a sequence of protein, you can predict how it folds. That's kind of amazing. And I think you can use that same technology now to predict how cells work, or you can use it to design new protein therapeutics. So the thing I'm most excited in the next 10 years is going to be the expansion of the use of AI to predict and to engineer biological systems, because I think that's going to dramatically increase our ability to understand how biology works, but also to manipulate biology.
请问林旭威同学在吗？麦克风要打开哦。
第二个做。
不好意思，刚刚没有听清楚。
谢谢你的回答，我大概能了解你所说的。
OK，Thank you。那我们再换下一个问题。再来是北科大机械系的谢承彦，是什么促使您创立 Synology、Tango 等多家公司的呢？因为您有波士顿最火红创业家的称号，让人非常想了解您创业的初衷。
对啊，其实 MIT 有个讲法，就是说 whatever you do as a student or as a postdoc or as a researcher or a professor, what they try to emphasize is impact, right? And you can make impact on the world in many different ways. You can impact the world by being a great educator, by teaching other people and giving them knowledge. You can make impact by publishing really great science and making discoveries. Or you can make impact by commercializing technology so that breakthroughs that happen in the lab now become real products for real people. So I think that is a really great philosophy to think about impact broadly. You know, what do you want to do with your life and impact to change the way things are done? When I started at MIT in 2010 as a professor there, what we realized, I spent the first 3 or 4 years just publishing research, and then what I realized is like that research was exciting, but unless somebody was there and we had more resources, it would never actually become a real product. So I had a bunch of great papers, but they never turned into something that real people could use. And so being in that ecosystem, I was very fortunate to be surrounded by other people from industry. So people from pharmaceutical industry, investors from the VC industry, other entrepreneurs who have started companies before. And what that allowed us to do is actually start companies where they would take that early technology and they would actually, see if it's useful. Number one, right? They have to confirm that it's actually useful. But 2 is then make a product out of it. So for me, I got to participate in these companies as a scientific founder. MIT allows professors to spend about 20% of their time, working on other things. And so that's what led to, starting Elego. Elego is building microbiome therapies for the skin. Synlogic is using probiotics to treat diseases in the gut. Tango is an exciting company that's developing new cancer therapeutics. These are all companies that hopefully, actually, they all have products now in clinical trials, and hopefully we'll have some real drugs that are approved. So yeah, I think the goal here has always been, to just take that next step so that these discoveries or platforms that we develop don't go to waste, that they actually, hopefully, become something useful for people.
OK， 那我们再换下一个问题。再来是北科大光电工程系的吕正豪同学。Funding and support. What types of projects in synthetic biology are currently finding it easy to secure funding and support?
So I think the ones that are the easiest today are probably ones that have to do with developing new drugs, because I think everyone recognizes that that's an area that has a lot of need. And the pharmaceutical industry has a lot of money to be able to invest in these things, and also VCs and investors are interested in this space. I think that's great for the field, but also not great for other applications of synthetic biology. So today I talked mostly about synthetic biology for human health, but there's actually a lot of other applications. I think one of the questions earlier asked about synthetic biology for agriculture, right? We could significantly improve food security in this world by engineering better crops. There's also ways of, for example, engineering cows to have, less production of greenhouse gases, right? So like, there's all these really amazing things you could do with synthetic biology in the food and agricultural space. But because of some of the challenges that the previous question asked about public perception, government, et cetera, there's been less investment in that space, which I think is a shame. The other areas that I think, synthetic biology can have a lot of impact on is in sort of climate change and environmental technology. So being able to make, better fuels, to make enzymes that can break down plastic. These are all things that biology can do very effectively. There's not as much research. There's not much research funding going into that space right now for a variety of reasons, unfortunately. So I think human health is right now probably still the area where it's easiest to get funding, both academic funding and industry funding to support. I hope in the future that, as these technologies get more and more mature, people realize the power of it for other applications.
请问吕
Thank
正
you
豪
for your answering, and I have one more question to ask about
Sure.
according to this one. It's like what factors are most appealing to investors or ending agencies when seeking funding for synthetic biology projects?
And so what was the beginnings? What projects or what topic? Or was it, can you say again?
What factors are most appealing to investors or
What factors?
lending-
Okay.
Yeah, factors.
Okay. Yeah. I'm less familiar with the academic funding environment in Taiwan, at least, but I would say in the US, for academic funding, I think this is probably true for all funding in academic, actually. You have to understand the audience first, right? So every funding agency has its own agenda. So, for example, if I want to fund something in human health, I need to understand what NIH wants in that space. Or if I want to fund something for the environment, I need to understand what the agriculture department or wants to fund in that space. So number one is like understanding what the other side wants is an important thing, because then you can shape your proposal there. On the commercial side, I think number one, the science is certainly important, but there's a lot of great science out there, and it's usually not the only thing that dictates success. One of the things I've found, two areas I'll find I think really key is the team. So and you want to try to create a team that has that interdisciplinary expertise. Some people who are great at the science, maybe they invented the thing at first, but you need to surround yourself with people who have been in industry before, who know the problems that are trying to be solved, or how to raise money, or how to build a team and sell companies. So building that team doesn't all have to be full-time. It could be advisors, it could be consultants. But really having that complete group is something pretty important. I would say the most important thing I've learned is that academic projects are not usually the best thing for companies to do, right? So what I mean is, when we published papers in the past, we were very excited. Like, we got this great paper, the PhD student can graduate. It's a great proof of concept. But usually as a professor, when I pick those projects, I didn't pick them really because I understand the commercial opportunity. I picked them because it was like the coolest thing, right? We thought it was awesome, so we can get it published. And so even though you have a great publication, doesn't mean that you should take that same exact thing you just published and try to commercialize it. Rather, I think it's important to step back and say, okay, now that I've demonstrated this, for example, now that I showed I could build this logic gate, what's the actual best application of this if I want to make this into a real drug? And that requires you to go out and talk to people, talk to other experts, talk to your customers, talk to pharma, talk to clinicians, to understand like what is the actual need here, and create a product or a business plan that actually solves that problem. And it's not just because like I created a cool technology and I want to see it go out. It's more work backwards from the problem and then make sure that your technology actually truly is a better solution.
Okay, thank you, Professor.
Thank you. 那我们再换下一个问题。
Yeah, what aspects of education?
education
Yeah.
training. What aspects of education do you believe need to be emphasized to cultivate the next generation of synthetic biologists?
Yeah, I think this is actually a broader question. I mean, this is like, what is the basic education anyone should have to get into this space right now? It's a good question. I think what I've realized is actually two things are most important in this space in terms of like actual coursework. But then there's a separate thing, which is the philosophy that one approaches problems with. So one is just to have a basic understanding of biology training, especially, the terms and the concepts underlying molecular biology. I find, again, I'm not as familiar with the educational system in Taiwan, but I know certainly in the US, we oftentimes encounter, college students who have never even taken a class of biology before. So like the concepts or the terms, they don't understand. They have to spend a lot of time figuring that out. So it's not too hard, I think, to take, online class, go to Coursera, go to MIT OpenCourseWare or something like that, and just take an intro to biology course. So you know the basic concepts, the languages to use. The second thing is I find that, a lot of people who get into the life sciences space, traditionally, maybe they did it because they didn't like doing math or quantitative technology, things like that. And as a result, they might not always have the best comfort with, computation or mathematics. And again, I'm not saying that one person needs to do everything, but you need to have some basic understanding of the approaches that people are using in the computational space, because I think it's going to be like one of the central things that's important in the future. So I would encourage people to, again, like take an intro to Python or intro to computer science course. It's not that difficult if you're willing to spend the time, and there's a lot of free resources out there to learn that. So those to me are like the two foundational things: learning the basic language of biology and learning computational. And then after that, you can decide, do I really want to specialize and go deep into computation or the biology or genetic engineering side? I think that's all later choices that one can make. I think the second thing is just like a philosophy about this approach. I think I mentioned this earlier. It's really about trying to solve the problems. The biggest challenge in this space is really identifying what the problems are, first of all, defining what those problems are very clearly. And then from that, I think the approaches will naturally come, right? So if you look at cancer, I think the one thing that we identified there is the central problem in cancer is, how do you tell the difference between a cancer cell and a healthy cell? That's the basic problem everybody's been trying to solve in the last 100 years of cancer treatment. So from that, you can work out different solutions. You have the same problem in other areas of biology. So identify that problem first and then work backwards, and be open-minded about, the approaches you're going to take to solve that.
OK， 请问吕正豪同学要追问问题吗？
No, just thank you, Professor, for your answer. I have benefited a lot.
OK， 那我们再换下一个问题。英文2的吴伯贤同学 ，Due to financial progress, the progress of the liver cancer CRNK therapy center 301A is delayed. Do you find a solution to continue the progress?
We did find a solution. Yeah. So actually, this is one of the interesting things about running a company now that you don't really think about as a professor, right? You oftentimes you have to manage complex budgets, and changes in the financial market can significantly change your plans, right? The stock market has changed a lot in the U.S. over the last 3 years, and it means that we have limited resources. So earlier this year, we announced with our liver cancer product that we were not going to develop it ourselves. We just announced a partnership last week with a company in China where they're going to develop the CAR therapy in China first. The benefit of doing that is that actually in China, you can do cell therapy trials very quickly. It's still pretty good, actually, quality as well. So we did a partnership. The partnership is worth about $150 million total. So they're going to do the first study there. And then if it's successful, they have the rights in China, but we have the rights in every other place outside of China. So I think it's a good partnership. It allows us to continue to develop that program, get data, but we don't need to spend money on it for now.
Thanks for your
OK，
Thanks
那
for
我们
your
再
answer.
换下一个问题。还是北科大机械系的谢承彦，您是在电机工程和生物工程2个领域中都有所建树。 想请问您是带着什么样的态度或是理念来作为在这条路上的前进动力呢？
Yeah, I think my main motivation in this space is really just motivated by the questions. Like I just fundamentally am interested in how biology functions. And I don't think anyone really understands it at the heart. Like how do these genes lead to cell function? Like no one really understands that. And so I think to work backwards from that problem, how do we solve it, right? And my approach personally is that we need to solve this through engineering. Other people have different approaches, but my approach is like, for example, if you gave me a car engine and didn't give me the blueprints and I had no idea how the engine works, I need to take it apart and I need to put it back together again before I can really say I understand how it works. And so that's my same philosophy in cells. Like I think for biology, I need to be able to take it apart. I need to recreate it. I need to make it better or worse. And if I can do that, then that teaches me that I now know how biology works. So it's really that curiosity of understanding how life works that's leading me to sort of use these sort of approaches. Again, everyone has a different philosophy on how they approach it, but that's my viewpoint on a very interesting way to understand how life works.
麦克风 OK， 麦克风没有开。
Hello.
OK， 有声音了。
Thank you for your Thank you.
好，没问题。
好，那再换下一个问题。来是北科大资材所的张兆仪，请问您拥有双重博士学位，分别是麻省理工学院 MIT 电机与生医工程博士学位和哈佛医学院医学博士学位，跨了工和医两个领域，为何不会有跨领域不专精的问题？以及您是如何分配读书、做研究、创业时间，才能拥有这么优异的成就？一般的学生光读一个科系都可能会有课业压力大的问题，请问您是如何跨这么多领域又能够这么专精的？我的指导教授说我跨领域不够专精，为何您跨的领域比我多，却能够如此的专精？
Good question. I think when you pick a career, I think you have that choice. Your choice is either to go very deep in one area, or your choice is to be broader, but try to have enough expertise across areas that you can work in a team and lead a team or work as part of a team to execute on an exciting project. And I think it's a personal choice for people on which one you want to go towards. There's nothing wrong with being very, very specialized and being the world's expert in that one area. I think, so for me personally, I have found it more interesting to try to integrate multiple approaches together. I admit I'm not the expert in every single space, right? So in the paper that we published in the Science and Nature papers on the electronics, I didn't do the electronic design, right? I found a very good lab that's the world expert in low power microelectronics, and they designed the chip, right? But I had enough ability to tell them and understand what they were doing to say like, look, this is not going to work because it's not going to work in the body, or it's not going to interface with all of my cells. So I think it's really a philosophical choice on what you want and what your personality is and what you want to do for your career. You can either go deep, or alternatively, if you're not as deep in one space, you need to have that language and the ability to pull together teams together to work together on projects. So I think that's how I personally approach it. So my philosophy has always been, let's try to increase the types of people I can work together with to solve these problems. For example, if you're a mathematics major, I think you should just be really, really good at mathematics if you want to solve the unsolved problems in mathematics, as an example, right? Because those sort of problems are not really typically solved by teams at this point. But for a field, and so this depends on the problem. Like the field of biology, I think is so broad and does require such interdisciplinary coordination that I think this is where having breadth is very helpful.
OK， and 这是我问的问题。那我也很想追问，就是所以是因为有兴趣，所以你才能够这么的 跨这么多的领域。那因为我是对什么都很有兴趣，可是什么又不专精，所以想说，到底要怎么样才可以，就是像您这样，哇，跨创业，然后又能做研究，又能够把书读得这么好，都是常春藤的名校。
这个主要是就是要选一个有兴趣的地方。我觉得说，if you pick a topic that you're excited about, it doesn't feel like work, right? So I think if you pick a topic you're not excited about, it's a lot of work. It's probably really painful too, right? So I think, you know, especially people who are in college, you know, obviously in high school, middle school, you have to learn the basics, right? But after that, you should think about really what you want to do, right? And I think once you pick that, it just makes it easier on yourself personally in your life to justify why you're spending so much time doing this. I think that's my recommendation. So that's hard to answer. That's hard for me to answer for you. I think everyone needs to think about like what is it that they actually care about.
Okay, thank you.
No problem.
那我们再换下一个问题。再来是暨大学资管的张祖林，合成生物学如何影响医疗领域？它有哪些潜在的医疗应用？
Yeah, I think right now the most immediate impact will be for cell therapy and gene therapy and CRISPR editing, because those drugs are going to get approved pretty soon, and there's going to be many, many drugs in the future. So, for example, I can see, 10, 20 years, I don't have cancer now, but at some point I'll probably develop cancer because everyone develops cancer. Hopefully when I develop cancer, there's going to be a product out there that's very effective, and it's probably going to use a cell or some more complex thing than just chemotherapy, maybe a more complex protein than chemotherapy. And I look forward to that day where we have much more precise drugs, so that when I go to the doctor, it's not like, okay, here's the thing that everyone is using and see if it works for you. No, it should be, I go to the doctor and they say, okay, this is what your disease looks like, because now I know the DNA and I know the information about that disease. Here's the best drug for you. And we've designed this drug to solve that problem. I hope that's the future, and I think we're going to be there. I think other areas that are very exciting, but a little bit further behind in this space right now are going to be things such as, trying to solve even more complex diseases. So for example, we don't really know how the brain works. No one knows how the brain works, really, right? And so part of the issue is we don't have a good way of engineering neurons. We don't have a good way of engineering a brain. But there's people now who are developing those techniques. There's a whole group of great, exciting people who develop this idea of optogenetics, where you can use light to turn on very specific neurons in the brain. And you can say, okay, if I turn on this neuron, what happens to the behavior? Or I turn off this neuron, what happens to the behavior, right? So we need those sort of technologies to understand how the brain works, number one. And then once we do that, we can hopefully build better drugs for brain diseases. So I think that area, which is like a black box right now, is, I think, really exciting. The other exciting area that I'm personally very interested in, why we developed the gut sensors, is we actually don't know what happens inside of our gut either. We can't see inside of there, but we know the gut affects so much of our body, right? It affects our health, it affects our happiness, it affects, our digestion, everything, right? Is affected by our gut. We don't really know how it works. So we need better ways of engineering the gut, or diagnosing the gut, or just monitoring what the gut is doing at different places at different times.
请问进大学资管系的张祖林同学在吗？
有在。
请问有要追问问题吗？
没有，谢谢。
好，谢谢。那我们再换下一个问题。再来是静怡台湾的周维珍，记其生物科技对我们生活最可能造成的影响。
Yeah, like I said, 已经造成影响就是那个 mRNA vaccine, because I think almost everyone has gotten it, right? Hopefully. So that already has a very direct impact on your life. I think other direct impacts, I hope none of you guys have any serious diseases, but if your family members have a serious disease and they go see a doctor, I think as of today already, there are drugs that were discovered using these sort of technology, or engineered using these sort of technologies. So I think you might be treated with one of those products, or your family members might be treated with one of those products in the near term. So I think these are some of the really key tangible things that are happening already with this technology today. There are other things that are going to be happening outside of, for example, actually, this is a good example. If your laundry detergent, 洗衣机, right? Like the reason why that laundry detergent is so much better now than it was 10 years ago is there are engineered enzymes inside of that laundry detergent. So that's already happening, right? You can clean your laundry better. So something as simple as that, that's enabled by genetic engineering. There's all sorts of new materials that are being made for clothes. For example, we can make materials out of spider silk. We can make bacteria and yeast produce silk, spider silk, and make materials that are much stronger than, or much better than the existing materials. So those sort of things are going to start impacting us, even though we don't really know that it actually comes from genetic engineering in many, many cases.
请问静怡台湾的周伟正同学在吗？3、2、1，那我们再换下一个问题。再来是北科大资材所的张兆仪，如何把电路板放到细胞上，让细胞像计算机那样具有记忆性，可侦测体内变化，然后做出决策、治疗疾病，或使用 TSP 最短路径技术。很好奇如何把半导体和基因组合在一起。
我们其实没有直接把电路这种东西放进细胞里。We're not putting electronics into cells. 可是我们是用就是 electrical engineering 这种观念去改变那个基因。So for example, the idea of memory, right? We're not using a memory circuit in semiconductors is basically using magnetic storage or transistors or capacitors to try to do that storage. We're not doing that, right? Because cells are not electrical in most cases, but we're using DNA in a very similar way. So it's taking the concepts from electrical engineering and using it to engineer biology, but we're not directly using electrical signals. We're actually using protein signals, DNA signals, RNA signals to do the same type of computation. So just like your brain, it's not silicon, but it still can do computation, right? Your brain has a different way of computing using, electrical spikes. We're using cells and DNA to do computation as well.
OK， 谢谢博士。那我们时间也差不多了，那我们最后一题就结束喽。那感谢听路与我们分享生物编程的历史，然后大数据及与人工智能结合正在革新生物学，让我们知道电子系统的能力呈现指数的增长，也认识 DNA 的读写技术，加速现代合成生物学的最新进展，以及将这些技术推向现实应用所面临的科学和商业挑战以及机遇。那我们请听路送给观众一句话。
Yeah, 非常谢谢大家的时间。Thank you for all the questions. I think hopefully this inspired some thinking in your mind. Happy to always chat more. So yeah, it was a fun conversation. Feel free to reach out if I can help with anything.
OK， 谢谢听路。那我们现在呢要来宣传11月30日到12月2号在南港展览馆2馆举办的 Meet Taipei 创新创业嘉年华。有请影片。
规模感觉跟过往比就更大了。
就今年有很多国际团队回来。
Just seeing all the incredible activity and energy with startup founders, that really connects to my being as being a former startup founder myself.
今年应该说在规模、在国际化以及在生态圈完整程度上，我觉得都取得很重要的突破。我觉得最大的帮助其实是譬如说一些顾问公司或者是新创业者之间的互相的交流，或者是一些资深的前辈给我们的回馈跟提问的建议，我觉得这边都非常的受用。 那密台北让我们大家可以聚集在这边，可以互相做交流，然后分享近况，其实是一个很棒的事情。
今年有非常多的投资人会一起参与这次的盛会，那可以帮助我们接触到更多国外的VC， 那未来可以更有效地扩展海外市场。
So I was incredibly impressed at what's happening at this conference. It is really extraordinary. The number of good startups, the excitement around opening up, the number of people who are really ready to go, break out and do something extraordinary is really something here.
我们希望这个创业家嘉年华也慢慢能够走向国际，不是只是在花博，我们希望是整个城市就有点像 Web Summit， 能够对这个城市带来很重要的影响。
那很期待是说，在第10年的 Meet Taipei， 其实我们也可以把这个品牌跟着这些新创团队一起往海外去发展，把它变成是一个国际品牌。
OK， 那今年的话是2023年11月30日到12月2号在南港展览馆2馆。那这是这次的嘉宾，那是礼拜4到礼拜6，欢迎大家来体验新创的感觉。好，那我们有请 NASA 喷射动力实验室的刘登凯博士为我们公布这次的得奖名单。
我先感谢这个我们的演讲的这个听录，真的是带来一个震撼的消息。每天这个看到这个 bio 这个进步啊，真是有点，我说比较普通的话，吓死人了。OK， 但是在吓死我之前，他能够发展一些医药想办法救活 ，That's what I'm looking forward to. OK，so 我想我们同学们问的问题都非常好，真的是非常好，而且非常投入的。我想Tim会 agree with me about that part. So it's a very difficult selections here. 所以第一个我先选这个我们是事前的问题。第一个我选择这个静宜台文这个周维珍同学，说近期这个工程对我们的生活可能大的影响。我想这个我们刚刚 Tim 给你回答了，所以我想给他这个第一名啊，就是1000块钱。第二名是这个北科大的谢承德同学。你问的是如何使用这个工业半导体的这个 IC 的观念，我看了一下他听的一些背景，我没有了解到，你提出来非常感谢你啊。OK， 所以这个第二个是500块钱。第3名是这个静宜的资管系张祖林同学，合成生物对如何影响医疗领域，我想这个 Tim 讲的很多了，非常非常不错的问题的，所以这个是一个。第一名是周维珍，第二名是这个谢承德，第3名是这个张祖林。那这个现场的问题的话，你稍微等我一下，我找的我的笔记本呢，稍微等一下啊。第一名是我选择了这个北科大林旭威同学，生物学的未来的发展，那讲的 AI 啦，这个我想 Tim 回答这个。让我就觉得这个我们同学们将来的可以发展东西非常多的，所以我就给这个林旭威同学第一名。然后第二名的话是我们谢承彦同学，这个第二次得奖。什么促使您创始这个一些公司啊？那我觉得我学到一个东西是 MIT 所说，impact the world. I love that, so I'm going to take it to heart. Choose my career to impact the world. I'm working on the NASA. I think that's part of it. 这是第二名。第3名是我看啊，第3名是这个北科大的吕正豪同学，他说是有关这个这个 real world application 这个实际上怎么应用，我想这个是非常好的问题，我也学了非常多。所以我们一共是6位同学，好吧？请6位同学都在这个我们的网上面来办理登记的。那我就先把这个现场交给我们的主持人赵毅同学。
好， 谢谢刘博士，恭喜以上的获奖同学，请得奖同学透过荧幕上的 QR code 进入得奖的LINE群，确认进入得奖LINE群或是有联络上的信箱再离开哦，以利奖金颁发作业。大胆提问才能成就非凡的未来。我们12月份演讲由 Ian Chen Music 个人工作室陈彦竹作曲家、音乐制作人演讲从乐谱到荧幕，揭开好莱坞音乐神秘面纱，以40闻名电玩原声带荣获第18届美国独立音乐奖 In Unnatural 短片在坎城国际创意节获得金牌。那再次感谢听录本次与会的学校同学以及来宾们，欢迎大家填写回馈表单，让我们能够做得更好。最后在这里也要感谢本次活动中参与幕后筹备工作的师长同学们，欢迎师长同学在我念到您时可以挥手，让大家来认识您哦。感谢大同大学的何明果校长，感谢电算中心包仓龙主任，感谢课程李尚林老师，感谢录影于佩轩，感谢与我们合作的辅英大学陈义谦老师，感谢动物大学秦玉国老师，感谢静宜大学陈明楼主任建慈老师，感谢辅仁大学周德莲老师，感谢文化大学周宗义老师，感谢实践大学朱良芳老师，感谢民传大学网络电视转播，感谢慈济大学张允志老师，感谢台北科技大学秦先玉老师，感谢嘉义大学陈明宇老师，以及感谢本次的宣传同学林小丽。感谢一大早不断跟各位确认设备状况的黄玉祥，谢谢大家今天的参与。大胆提问才能成就非凡未来。我们12月份演讲再见喽，谢谢，拜拜。
那个赵